Uh, Thank you very much. Ladies and gentlemen, good morning. So, transfusion-related acute lung injury is the leading life-threatening risk of any transfusion. In a recent systematic review, we've shown that red cell transfusion in cardiac surgical patients is associated with a twofold increased risk of developing TRALI. Moreover, TRALI is influenced by red cell storage duration. So this is this famous chart from the landmark paper by the Cleveland Clinic, which shows that older blood is associated with worse outcomes. In a porcine model of transfusion-mediated organ injury that we recently developed, we've shown that TRALI is associated with platelet activation. So the objective of this study was really to evaluate whether removal of the red cell storage lesion by washing will ameliorate or prevent TRALI. So specifically, we wanted to look at the effects of washing on the quality of stored porcine erythrocytes to determine whether removal of their storage lesion using washing will prevent TRALI in the porcine model, and further characterize the effects of transfusion on platelet function and coagulation. So this is a schematic of our model. We used adult landrace pigs, put them under general anesthetics, had 3 time point measures at baseline, 1.5 hours and 24 hours post-intervention, where we assessed lung function and took bloods for further analysis. 24 hours, we also took bronchoalveolar lavage specimens and harvested organs for further analysis. We had 4 groups: a sham group that received saline, and 3 transfusion groups. The transfusion groups received either day 1, day 14, or washed day 14 red cells, and all transfusion pigs received about 1 liter of leukodepleted cross-match allogeneic red cells. For the washing procedure, we use this Fresenius CAT system, which is present in many of our units. And these are our results. So firstly, with regard to the in vitro work, washing, we showed ameliorates the red cell storage lesion without altering the quality of stored red cells. So if you look at this column, which is washed day 14 red cells, we can see washing preserves hematocrits and erythrocyte ATP and 2,3-DPG concentrations, but reduces the increases in potassium and lactate. Washing also preserves erythrocyte deformability and preserves the 24-hour in vivo survival of these red cells once they're transfused. We also showed that washing reduces the microparticle load or burden. So this is just a graph of our flow cytometry analysis where we labeled for annexin 5 and showed that washing reduces the number of microparticles compared to unwashed cells. Secondly, we showed that washing ameliorates TRALI but not pulmonary hypertension. So in all these graphs, you'll see that the red bars or lines are day 1 red cells, the blue ones are the day 14 red cells, and the green are washed day 14 red cells. And washing ameliorates this reduction in arterial oxygen tensions over time, with a similar effect on histological lung injury scores, where it reduces histological lung injury. And we can see this on this H&D section. So unwashed day 14 group has this proteinaceous debris in the alveolar space, hyaline membrane formation, and neutrophils in black arrows. And this is ameliorated in the washed group. However, transfusion of aged erythrocytes, irrespective of washing, causes pulmonary hypertension, assessed here using a Swan-Ganz catheter. We then showed that washing ameliorates blood and pulmonary platelet activation and inflammation. So these graphs are of whole blood analysis using flow cytometry or ELISAs for IL-6, and we showed that washing reduces the expression of platelet activating complex and P-selectin in blood, and it reduces the release of IL-6. A similar effect is seen when we look at lung tissue. So these are immunofluorescence images from porcine lung, and if we look here, there's increased activated platelets in lung tissue, which is reduced in the washed group. Similarly, with macrophages, there's reduced infiltration of macrophages in the pigs that received washed red cells. Finally, we showed that red cell transfusion causes platelet dysfunction and coagulopathy, which is aggravated by storage and unaffected by washing. So in this graph, we showed that transfusion of aged erythrocytes reduces plasma platelets out to 24 hours, irrespective of washing, and it also causes platelet dysfunction. So platelet function was assessed by multiplate, and specifically here, we looked at platelet aggregation in response to ADP. And we can see transfusion of aged erythrocytes causes platelet dysfunction, which is not affected by washing. Coagulopathy, we assessed using ROTEM, and this is just from our XSTEM arm, and we show that, again, transfusion-aged erythrocytes causes increase in clot formation time and reductions in maximum clot formation. Interestingly, there's these trends where day one red cell transfusion also causes mild coagulopathy, so mild increases in clot formation time and reductions in maximum clot formation. So in conclusion, ladies and gentlemen, any allogeneic red cell transfusion causes pulmonary dysfunction, coagulopathy, and inflammation in this model, but to differing degrees. So old red cells cause worse pulmonary dysfunction and coagulopathy. Washing ameliorates this pulmonary dysfunction, but does not totally eradicate it. Must also remember that red cell transfusion is also beneficial. So graphs I haven't shown, it increases hematocrits and increases global oxygen delivery. And our next step is really to conduct a randomized controlled trial of red cell washing cardiac surgical patients, which has already started actually. And secondly, we need to determine what the changes in the stored red cell membrane are which lead to this coagulopathy and inflammation, and see if we can target this therapeutically. And the last thing is, I think it's important to ask the question, what indicators determine when a transfusion is beneficial or harmful in patients? I'd just like to thank our funding bodies and the numerous people involved in this research. Thank you.